Hello, it's Miss Bell again. Today we're going to be talking about chromosomes and cellular reproduction. So here you have a really cool picture of chromosomes, and I want you to notice how there are different sizes and shapes, but you have some that are similar sizes. We're going to be talking about that in a few short seconds. But anyway, so this unit is on how cells reproduce and the role that chromosomes play in that. And if you recall, chromosomes are DNA, and so of course DNA has a central role in reproduction. So the next phase in the cell cycle is mitosis. So after interphase, you'll have mitosis. Mitosis is much shorter. It lasts only about 1 to 2 hours. Mitosis is only about 5% of the total cell cycle. So what mitosis is the nucleus dividing, or nuclear division. Like I said earlier, the mitosis does not actually divide the cell. Okay, so anyway, this is the nucleus dividing out. So this is in somatic or body cells. So every cell in your body except for sex cells. So if you get a cut or you break a bone, all growth, development, and repair is through mitosis. So one of the first things that has to happen is going to involve DNA. And DNA in interphase is chromatin, remember? Okay, so first chromatin has to become chromosomes. So the first few steps in mitosis is going to involve splitting out the nucleus. And then the last step of mitosis is going to start splitting the cell and reforming the nucleus. And then your last step of the cell cycle involves splitting the cytoplasm, which is cytokinesis. Okay, so remember your 3 steps to the cell cycle are interphase, mitosis, then cytokinesis. So in mitosis, the nucleus divides, and then in cytokinesis, the cytoplasm divides. So at the end of mitosis, you should have 2 clones, 2 identical daughter cells that are diploid, okay? So you will have 2 diploid somatic cells. So you take 1 diploid cell and it becomes 2 diploid cells. And they are considered clones because they're genetically identical. All your skin cells should be exactly the same, right? So anyway, in mitotic cell division, the parent cell, so your 1 diploid cell divides to form 2 diploid cells. So you start with the same number of chromosomes you end with. So no haploids in mitosis. Mitosis only deals with diploids. So for example, if a human cell has 46 chromosomes, at the end of mitotic or after mitotic cell division, it will still have 46. Because if you recall, you start with your diploid with 46. Then in interphase, your chromosomes double to 92. And then at the end of mitosis, you have 2 that splits, and then you have 2 that are 46 each. A frog cell has 26 chromosomes. After the first mitotic division, the cells produced by the zygote will have. Okay, so if a frog cell has 26 chromosomes, it doesn't tell me what type, but we're going to assume that's diploid. And then, so after mitosis, a zygote is also going to be diploid, by the way, because sperm plus egg makes diploid. Anyway, so it should be the same number, 26 chromosomes. So the 4 stages of mitosis are PMAT: prophase, metaphase, anaphase, telophase. Okay, so this picture shows mitosis, and it starts out with interphase. So if you notice, this is actually the cell cycle. So if you notice, we're going to put this as a starting point. Okay, so if you notice, you have 2 cells formed here, right? Okay, so they're going to each begin the cell cycle over again. So here's interphase. So notice you don't have chromosomes. DNA is kind of just uncoiled in chromatin. And so in interphase, that's where you go through G1, S and G2. So the cell grows and doubles its DNA to 92 chromosomes. And then we get into prophase. Okay, so both of these phases could be considered prophase. Okay, you have early prophase and then a later prophase. So in prophase, you'll notice the DNA goes from chromatin to chromosomes. That's one of the first steps. Also, notice the solid line here for the nucleus, and then it becomes a dotted line because the nucleus is breaking down so that the chromosomes can move to opposite sides of the cell. Another thing that's happening is these centrioles here are moving to opposite sides of the cell because they're going to help pull the chromosomes. So if you look here, they're pulling the chromosomes to opposite sides of the cell. So anyway, and then those spindle fibers are starting to form. Okay, so those are the little lines coming off of the centrioles. So the spindle fibers, see the developing spindle? Another thing you might hear centrioles called is an aster. Okay, so centrioles may also be called the aster. And then you get to metaphase. So this is kind of like the cell going into metaphase. So those spindle fibers begin moving the chromosomes to the middle. So this one is like them moving, and then now they have reached the middle. So metaphase is when the spindle fibers have moved those chromosomes to the middle so that they could easily be separated. Okay, so again, here's metaphase. Okay, and then we're going into anaphase here. Okay, so if you look here, the X's look different because they're starting to be split into their sister chromatids. And then here, the chromatids have split and they begin moving towards opposite sides of the cell. So they're moving towards those centrioles. The centrioles kind of act like a fishing pole, and they're reeling in the chromosomes. And then in telophase, here your chromosomes reach opposite sides of the cell. And so then the spindle fibers don't need to move them anymore. And so spindle fibers start going away. You'll notice also that your centrioles are still on opposite ends. And in telophase, the nuclear membrane will reform. And then at the end of telophase, the chromosomes will go back to chromatin. And then here we have cytokinesis here, where you actually split into two new cells. So if you noticed, mitosis is the nucleus dividing, and then cytokinesis is when the cell finishes splitting. Okay, so this is just a simplistic picture of mitosis. You have your parent cell, which has 46 chromosomes. DNA replicates during the S phase of interphase. So it replicates to 92, and then my two daughter cells each are diploid. They have 46 chromosomes. That brings us to the first step of mitosis, prophase. Okay? So prophase, I've underlined some key things. So the things that I've underlined would be good things for you to highlight. Okay, so they are the most important things that happen in each phase. So prophase, chromatin becomes chromosomes. Remember, chromosomes are much easier to see. And actually during prophase, this is the easiest time to see the chromosomes because the DNA has been shortened and thickened. If you think of a ball of yarn versus yarn being stretched out. If I was to stand across the street at the Votech or the post office, and I was to put someone over there, and then I was to stand in the parking lot, if you had a ball of yarn, I could probably see it because it's shortened and thickened. But if you were to stretch it out into just one string, it would be much harder for me to see across the street because it would be like more thin and stretched out. So I kind of think of chromosomes like that, except you're looking through a microscope. Anyway, so chromatin becomes chromosomes. So chromatin kind of is all squiggly, and they become X's, your chromosomes. So each chromosome has been duplicated. It's made of the two halves called the sister chromatids. And so that's a replica of the DNA. So anyway, the other thing that happens is that the nuclear membrane disintegrates or goes away or disappears. And this can be represented by a dotted line in prophase. Spindle fibers begin to form between the centrioles. If you recall, centrioles are those organelles that are only in animal cells, and they are paired and usually close to the nucleus because they play a role in cell division. Okay, so we're going to talk about that role now. So one thing that that centrioles do is they help form these spindle fibers or spindles. Spindles are little protein type structures made up of microtubules, and spindles move the chromosomes during cell division. They help move the chromosomes so that it can split into two new cells. So centrioles help make the spindles. Now another thing that happens is that centrioles move to opposite sides of the cell. So again, those centrioles are little organelles. Again, they're made of protein, microtubules, and they're only found in animal cells. And the centrioles are going to anchor the spindles as they move the chromosomes. Okay, so that's what they do. A pair of centrioles is called a centrosome. So if you see the word centrosome, that is the pair of centrioles. And again, the spindles are going to pull the chromosomes towards opposite sides of the cell. So the centrioles go to the opposite sides of the cell. They form the spindle as they're doing that. And then those spindles during metaphase are going to grab a hold of the chromosomes, and then they're going to move them towards that anchor. They're going to pull them towards that centriole. So if you take a look, this is a cell in prophase. A couple things I want to point out. Notice the dotted line because the nucleus is disintegrated. Okay. Notice the centrosomes, the pairs of centrioles, are beginning to move towards opposite sides of the cell, and your spindles are forming because eventually those spindles are going to grab a hold of the chromosomes. And of course, lastly, we have X's. So chromatin became chromosomes. Now it depends on what phase or what part of prophase you're looking at. If you're looking at early prophase versus late prophase, sometimes you might see DNA represented as chromatin. It just depends on what point of prophase you're in. But during prophase, do know chromatins become chromosomes. So again, as each pair of centrioles goes to the opposite side of the cell, they'll pull one set of those microtubules or one set of the spindle fibers, and then the other set is going to connect to the chromosome, the half of the sister chromatid. Where that spindle fiber attaches onto the chromosome, onto the centromere, is called the kinetochore. Okay, so if you look at the centrioles and their microtubules, you'll see that they're very similar under a microscope. And we don't have microscopes that can show that at our school. Anyway, so the asters, again, the appearance of those two asters with their microtubules coming out, that's known as the mitotic spindle. So it's basically the same thing. Anyway, so chromosomes are very distinct and they're very easy to see in prophase. So this is when most genetic studies are done. Okay, so like in metaphase, you'll see the cell, and I'm going to put the circle for a centriole. So they're on opposite sides of the cell. And so the chromosomes are lined up in the middle. And of course, you know, there's going to be 92 of them, so I can't draw that many. But so the spindle fiber comes out of the centriole, connects to the chromosome close to the centromere. So where it connects is called the kinetochore. Okay. And then what's going to happen, so they're all going to be connected. And what's going to happen is in anaphase, in the next phase, here, I'm going to just erase this so I have space. Well, let me do it over here. So then in anaphase, what happens is your centrioles are in the same place, but those chromosomes are split. Sorry for my drawing. So the spindle fibers are still connected, and they start pulling those chromosomes. They start pulling them towards the centriole. So start pulling towards that anchor, that centriole. And eventually it's going to split into two new cells. All right, chromosomes are most easily seen during cell division because chromosomes, do they double in number, shorten and thicken, move and expand, or match up with other chromosomes? Well, hopefully you recall the analogy I mentioned about holding the ball of yarn across the street. So chromosomes are easier to see because they have been shortened and thickened compared to chromatin. Which of the following does not occur during interphase? Chromosomes become visible, proteins synthesize, chromosomes duplicate, or proteins translocate? Well, hopefully you remember chromosomes are never visible when they're chromatin during interphase. So that brings us to the next phase of mitosis, which is metaphase. Metaphase is usually one of the easiest ones for people because it's very simple. Metaphase is when they line up along the equator or middle. So chromosomes line up along the middle of the cell. So again, you can see your centriole here. Let's change to red. You can see your centrioles here, and your spindle fibers are coming out, and they've attached to the kinetochore, and they have moved those chromosomes to the center of the cell. Those spindle fibers have helped move those chromosomes to the center of the cell. And if you look here, we've got some cells going through metaphase. Which phase of mitosis is represented by the diagram shown above? Hopefully you picked metaphase because it's lined up in the middle. All right, then you have ana. Ana means across. So in this phase, the spindle fibers are going to continue to move the chromosomes. But what they actually did was they split them. They're not X's anymore. Now they're V's. If you split an X, you get two V's. So the spindle fibers have pulled the sister chromatids apart of the chromosome. So now they're called chromatids. Okay, and those chromatids are going to move towards opposite ends. So here's your centriole, your anchor. Those spindle fibers are shortening, reeling in that chromosome towards the anchor, the centriole. And eventually they're going to pull them all the way in. Okay, and then eventually we'll get to new cells. Again, you can see that occurring here in the plant cell. You can clearly see that this is going to eventually become two new cells. So then the last phase of mitosis is telophase. So one thing I want to say about telophase is it's the opposite of prophase. Okay, so instead of one nucleus disappearing, you have two new nuclei forming. Okay, so again, it's the opposite. So in prophase, your chromosomes are in the middle. So your identical chromatids have reached opposite sides of the cell. Then they're going to uncoil into chromatin. So in prophase, chromatin became chromosomes. In telophase, chromosomes becomes chromatin again. Okay? The nuclear membrane disappeared in prophase, but it's going to come back. See the dotted line? It's coming back. It's reforming. But except there's two of them. So that's the opposite as prophase. In prophase, spindles appeared. Now they're going to disappear. Anyway, so you can see very clearly that two new cells are in the process of forming. The nuclei are reforming. So the first three steps of PMAT, of mitosis, prophase, metaphase, anaphase, you split the nucleus. In the last step of mitosis, telophase, you're putting the nucleus back together. You are not splitting the cell yet. That happens in cytokinesis. Remember, there's three parts to the cell cycle: interphase, mitosis, cytokinesis. Okay, so mitosis is the nucleus dividing. Cytokinesis will be when the cell actually finishes dividing. So after mitosis ends, cytokinesis begins. All cytokinesis really does is it divides out the cytoplasm and the organelles, and then you get two new cells. Okay, so notice you have chromosomes, your sister chromatids here, but then down here you have chromatin. Your nucleus is forming. You've got two separate cells. Your spindle fibers have shrunk up and went away, and like I said, your nucleus is reforming. So your new daughter cells that were formed in cytokinesis will then begin interphase to start the cell cycle over again. Which phase of the cell cycle involves replication of chromosomes? Remember, chromosomes are your DNA, so when is DNA replicated or copied? Hopefully you remember DNA is synthesized during the S phase of interphase. Following telophase, two daughter cells are created when new membranes are formed in the process called, well, after telophase, you should recall that cytokinesis is next, and that's when you get two new cells. All right, when chromatids have been separated, the cytoplasm is separating, and a visible line is forming between the sets of chromatids, which phase of mitosis is being described in the lab notes shown above. Well, if the cytoplasm has started separating and the chromatids have already been separated, that means it's telophase. The stages of cell division called PMAT, prophase, metaphase, anaphase, telophase, occur during which stages of the cell cycle? Well, hopefully you recall that G1, S, and G2 are all interphase. And M is mitosis. Mitosis is when PMAT occurs. Oops, sorry. Which event in mitosis has not yet occurred? Okay, so which event in mitosis has not yet occurred? Chromosomes lining up, spindle fiber shortening, chromosomes duplicating, or cytoplasm dividing. Hopefully this is an easy one for you. Eventually you can see that two new cells are going to form, so the cytoplasm has clearly not divided yet. So what are the differences between plant and animal cells undergoing mitosis? Well, there are a few key differences. Plant cells do not have centrioles. So all that we've talked about centrioles, plant cells don't have them. Plant cells have a cell wall. Okay, so they divide their cytoplasm with something called a cell plate here. Okay, you see it's like new cell wall forming here. So that is a cell plate. So plant cells have a cell plate. It eventually becomes their cell wall. In animal cells, they have what's called a cleavage furrow. Okay, so it's that pinching between the cells. And then as the cleavage furrow forms, that becomes the cell membrane. And it's going to form a contractile ring, eventually pinching off those. You can kind of see the contractile ring here. So it's pinching off to become two new cells. So this brings us to factors that control the cell cycle. So, obviously the cell cycle is very carefully controlled, but if it is not, this can result in cancer. So how you get cancer is when the cell cycle goes unchecked or uncontrolled. We're going to talk about a couple of different mechanisms that help control it. So cancer is the direct result of the cell cycle not being controlled. This control is primarily maintained by DNA and your genes. Your DNA, as you recall, gives the instructions to make certain proteins and also to regulate the cell growing and regulate the cell dividing. And it tells the cell when to divide and when to grow. So there's a couple different factors that help control this. First off is external signals. So I guess we could say factor number one would be external signals. One of the external signals of a cell, so these are things outside of the cell that help signal for when the cell should divide. Growth factors are one example of this. So these are certain proteins that signal the cell to divide or go through mitosis. And this can be produced by cells or released from damaged cells. So you might wonder why your bones don't continually growing, but if you break a bone, then they'll grow. Well, that's because when you break a bone, those damaged cells that were broken are going to release a growth factor. Those growth factors then bind to the receptors on the surface of nearby cells, and then they tell those cells you need to multiply to fix this problem, to repair this problem. And some are only going to attach to certain types of cells. So the second factor is going to be internal checkpoints. So once a cell is stimulated by the external growth factors to divide, then internal factors are then activated to make sure that the division goes correctly. So you have these checkpoints. So checkpoints are different points in the cell cycle where if something is wrong, there is a stop. It stops the cell cycle. So if something goes wrong, then the cell cycle will stop at that point until it can be fixed, or possibly the cell might just die. So checkpoints are made by these special proteins called cyclins and kinases. Those are enzymes, which of course enzymes are proteins. Now apoptosis can occur. Apoptosis, if you recall, is programmed cell death. So this happens when cells become damaged or worn out, and it can also occur whenever if the cell signals a gene to produce the digestive enzymes in the lysosomes. Basically, in the cells, the lysosomes will all burst at the same time for apoptosis to occur. So anyway, this is what can happen, like when in an embryo, so that they don't have webbed feet. Apoptosis occurs, and we've talked about that before when we discussed lysosomes. But this can also happen if the cell's going through a checkpoint and it cannot repair the problem. So your checkpoints. So there's three main checkpoints. The first checkpoint is the G1 checkpoint. Guess what phase that is in? G1 of interphase. So in the G1 checkpoint, again, this is in the G1 phase of interphase. So this is before the cell even begins dividing. This determines if the cell is big enough, if it's reached the right size, and if the cell has enough energy and nutrients to go through the cell cycle. The cell will also check for damage to DNA and make sure that there's enough growth factor signals for the cell to divide. Then the G2 checkpoint, of course, occurs during the G2 phase of interphase. If you recall, this is after the S phase, so DNA has already been replicated. This prevents the cell from entering mitosis if the chromosomes have not been duplicated correctly. So let's say you don't have 92 chromosomes. Let's say you have 93. Well, then hopefully it would prevent the cell from going into mitosis. Then you have the M checkpoint. This is the mitosis checkpoint, specifically during metaphase. So mitosis metaphase, MM, pretty easy. So this happens, like I said, in mitosis near the end of metaphase, and this checks that all your sister chromatids are attached to the spindle so that they can pull them to opposite sides of the cell. So when they do get pulled, remember in metaphase, your chromosomes are lined up in the middle. So it's checking that right here that it's attached. Okay, because if it's not attached, let's say you have one and these two are attached, but the one on the bottom is not, then this cell will have one too many, and when it divides, this one won't have enough. So it would look like this. So this one would have too many, this one would not have enough. So one unfortunate thing in life that humans have to deal with sometimes is cancer. So cancer is uncontrolled growth of cells. Specifically, it's a mistake in the cell cycle, okay, or cell division. As I've told y'all before, my brother, when he was 14, he was diagnosed with leukemia. Specifically, he had T-cell leukemia. And the T cells are a type of white blood cell. So leukemia is a cancer of the white blood cells. Most leukemias occur in the B cells. His was the T cells. So anyway, what happened is he had an overgrowth of immature T cells. If they're immature, they don't do their job. And then you get way too many of them, and they crowd out your blood. And when they crowd out your blood, the other parts of your blood do not work. So his platelets and his red blood cells wouldn't work, and all the other things. So anyway, a normal white blood count is going to be between 5 and 10,000. When he was diagnosed with leukemia, his was 169,000. So as you can imagine, he had way too many, and they were all immature. So he had no immune system at that point because he didn't have any mature white blood cells. He also had to get platelets and red blood cells transfusions because it crowded out those cells and actually killed them. Anyway, so my mom also had breast cancer. Fortunately, both my brother and my mom are doing great. But like I said, cancer is kind of a fact of life for humans. We're very fortunate that we live in a time period where a lot, but not all, of cancers are treatable. It's hard to say that there's like a specific cure because every cancer has different stages. And so we're going to talk about kind of just general how cancer works. So generally, cancer is that uncontrolled growth of cells. If you catch it earlier, then it's easier to control that growth of cells by killing the cancer cells. But what happens is, you have, if you have one cancer cell and it makes it through one of those checkpoints, you have one. Okay, then it divides, and then you have two. And then those divide, and then you have four. And at this point, you wouldn't know you had cancer. And then those divide. And you see what I'm saying? You have exponential growth. And so with cancer, it depends on the type of cancer. Some cancers cause symptoms very quickly. Some don't. Some cancers, they, are easier to spot. Some are harder to spot. It just depends. So anyway, if the cancer is caught earlier, in the division process, you don't have as many to kill and they're all in one spot, it might be easier to kill those cells. Ultimately, the cancer cells have to be killed, though, or they will take over. So anyway, if the abnormal cells are not destroyed at those checkpoints, so if your body doesn't destroy them, and you have all those checkpoints in place to prevent that, then internal growth factors are not suppressed, then you have possibly cancer, an uncontrolled growth of cells, and then they keep dividing and you get more and more of those cancer cells, like going from 5 to 10,000 to 169,000. They're estimating that my brother only had it for like 1 or 2 weeks. I want to say they had I think 2 weeks is what they told us. And so anyway, his was a faster growing cancer, of course, and my mom's, they caught it very early, and it was a very slow growing cancer also, so we were very lucky with that. My brother had to go through 3 years of treatments. The type of leukemia he had, they couldn't do a bone marrow transplant for him, so he had to just go through almost 3 years of chemotherapies and radiation and just all kind of stuff. Anyway, so why do people get cancer? Sometimes you get cancer because of a certain carcinogen. That's the easy answer. So carcinogens are things that cause cancer. Carcinogens damage the DNA in your cells, and that's how it causes the cancer. So tobacco, of course, is a known carcinogen. It's one of the top carcinogens in the world. So tobacco is going to primarily affect the cells that it touches. It could happen, it increases your risk of cancer anywhere in your body. But for example, if you dip, then it's going to primarily affect where you put the dip in your mouth. And then if you swallow the spit, gross, then it could affect your throat. But a lot of people that dip, they get some sort of mouth cancer. If you smoke, a lot of those people get cancer in their lungs. But you could get another type of cancer too. So tobacco just increases that risk. UV radiation, that's the sun. So of course, the sunlight, the more UV radiation you're exposed to, the more likely you are to get skin cancer. Of course, y'all probably heard of x-rays can cause cancer. It takes a very large amount of x-rays to cause cancer. Nowadays, the x-ray, and y'all know I used to work in x-ray, so I can tell you these things. Nowadays, one chest x-ray, you get less radiation than going on an airplane flight for 2 hours, because you're closer to the sun and you are receiving some radiation then. But anyway, you're receiving background X radiation from the earth. Depending on where you are, different places have more of it. We've talked about that in class before. Anyway, asbestos is a chemical type thing. It can be found in lots of places, but they have phased it out to where it's only in absolutely necessary stuff. It used to be found in like house insulation. So people that worked in that and didn't have proper protection on. Some viruses can cause cancer. If you've heard of the Gardasil vaccine, that protects against the HPV virus, which causes cervical cancer in women. And apparently a lot of people get it. A lot of people get HPV. If you get HPV, then it increases your risk of that cancer. So they've made a vaccine for that. You get it, I think you can get it in your teenage years. I don't know. It wasn't around when I was a teenager. And anyway, but another thing cancer can be caused by is aging and stress and environmental exposure to those carcinogens. So let's talk about aging and stress. So when you get older, things don't work as good. Your cells don't divide as quickly, and they could easily make a mistake. Stress also can cause different things to happen in your body, and basically stress can just make everything mess up. So those are a little bit harder to pinpoint why the cancer occurred. There are some cancers that are genetic, or certain carcinogens might mutate your DNA, permanently changing your DNA. Like I said, certain cancers are genetic. Those are caused by an oncogene. Onco is cancer. So this is a cancerous gene if it is expressed. So let's talk about that for a second. 20% of breast cancers are caused by the oncogenes, the BRCA1 and BRCA2 genes. I worked in MRI before I was a teacher, and we did breast MRIs on patients who had breast cancer every day, sometimes multiple times a day. And one of the things we were looking at in their chart was to see if they had genetic testing. When my mother was diagnosed with breast cancer, they tested her genetics to see if she had the breast cancer gene. And she did not. So because of that, I probably don't have it either. So that's a good thing. If you have the breast cancer gene, you have the gene when you're born. You have your DNA when you're born. One of my good friends growing up, her mom, all of her aunts, and her sister all have had the breast cancer gene. And so they've everyone but her sister has had breast cancer. Her mom and all of her aunts. And so her sister, because she did have the gene, she decided to undergo a mastectomy where they removed her breast before she got cancer. My friend did not have the gene. She's the only female in their whole family that did not have the gene. So anyway, if you have the gene, it doesn't mean you're born with it. It doesn't mean you're born with cancer, but you are born with the gene. And at some point, that gene could be expressed. Maybe it doesn't get expressed. Maybe you die in a car accident or heart attack before the breast cancer gene is expressed. But it's there, and it can be expressed at any time if you have the gene. So if you get extra stressed or as you get older, you're more likely for that gene to be expressed. So cancer is a very fickle thing. So when people say, oh, we want a cure for cancer, that's a very broad statement because cancer can occur in pretty much any cells in your body. Most cancers This is weird though. Most cancers are not going to occur, so it would be very rare for cancer to occur in your amitotic cells. Okay? Remember, your amitotic cells were going to be your nerve cells and your heart muscle cells. Now, does that mean you can't get brain cancer? No, that's not what that means. A lot of brain cancers are in the area surrounding the nerve tissue. In your head, you don't just have brains. You have things that line the brain, like the meninges line the brain, or like the glial cells. You have different cells in your brain that are not all nerve cells. So a lot of the times cancers that are occurring in the brain are going to be attached to that. Anyway, so it would be not as common for you to get cancer in those cells. And when you get into different types of cancer, then it just gets very complicated. But anyway, so cancer cells are going to affect your normal cells. They might form a tumor. So in leukemia, you're not going to have a tumor because blood is fluid. But you could develop a tumor if it metastasizes. With breast cancer, though, my mom had a very small tumor. Like I said, they caught it very early. The tumor could be benign. A cyst is considered benign. Cysts are usually fluid-filled, so it's not usually filled with cells. So tumors might be benign. If it's benign, it's not going to invade other tissues. Malignant means it's going to keep invading other tissues and destroy them. So wherever the tumor is, it's killing healthy cells. So those cells will not function anymore. Once cancer gets to the upper stages, and it depends on the type of cancer, how they stage it, but let's say you have like a stage 4 cancer, usually that's like an upper stage. Then the cancer cell, that usually means that it has gotten into your bloodstream, and then they have stopped somewhere else and formed a new tumor. And then it's going to also mess up the organs. So basically it's moved somewhere else. That's called metastasis, or it's metastasized. Example would be my grandfather. Both of my grandfathers had cancer, actually. On my dad's side, they believe that he had cancer from working, and he died years ago, but they believe that he had cancer. He had lung cancer, but he never smoked a day in his life, and they believed he got it from working in the shipyard, and he was working around asbestos, and he was exposed to it for like 40 years working with it, with no equipment, no mask or anything, and so they believe that that's where his cancer came from, was in his lung. They didn't find it until, because he didn't have any real symptoms, and then he had cancer, and it moved to his liver, and then it moved to his bones. By the time they found it, it was everywhere, and he only had 6 weeks from the time they found it, and then he passed away. My other grandfather, he had prostate cancer, and he went through some treatments that were pretty mild, never lost his hair or anything, didn't really get sick, and then they were like, oh, okay, you don't have cancer anymore. And then he went back for his checkup like 2 years later. I think he went like maybe every so many months. But anyway, it was about 2 years of not having treatments. And then he had cancer, and it was in his bones. And after it moved to his bones, it moved to his lungs, and then it moved to his brain, and it just went everywhere. So that's how metastasis works. And once it gets to all those other organs, you can't just kill all the cells in your body. How cancer treatments work is you can cut it out through surgery, you can radiate it, which is going to kill it, or you can do chemo, which is going to kill it. So obviously surgery cuts it out. You can't always do surgery though, because if it's in multiple places, you can't just cut out half of your body. With surgery, if it's around an important blood vessel, you're taking a risk there with killing you that way. And some doctors will or will not operate. It just depends. With chemo and radiation, they're going to kill the cancer cells. But when they kill the cancer cells, they're also going to kill surrounding healthy cells. So they can only do so much of that. When they kill those healthy cells, it's going to cause major side effects. One thing chemo does is it lowers your immunity because it's going to kill your white blood cells in your bloodstream because your blood goes everywhere. So no matter where you get the chemo done, it's going to kill those cells. A lot of chemo, they just do it in the bloodstream. It's like an injection. My brother had chemo every way you can have it. They did a spinal tap once a week where they pulled out spinal fluid. They put the big needle in his back, and they put chemo in that way. He had shots in his legs that went into his muscles. He had chemo that way. He took chemo pills. He took chemo injections. So anyway, and chemo is known for the nausea and the vomiting. So, but if it saves your life, it saves your life. So here is a picture of some lung cancer cells, and you can see they're kind of forming a tumor-like looking thing. So that's going to invade healthy tissue and make you not be able to breathe. All right, which of the following best describes the purpose of mitosis? So what is the purpose of mitosis? The production of a complete copy of the nucleus before cell division, division of the cell after the nucleus has been copied, the production of gametes with half the number of chromosomes of a normal cell, or the replication of DNA within the nucleus. Well, hopefully you recall that mitosis is making a new nucleus, okay, or the nuclear division before the cell actually finishes dividing. Division of the cell after the nucleus has been copied, this would be cytokinesis. Production of gametes, that would be meiosis, which is the next video. And then the replication of DNA within the nucleus, that's interphase, specifically the S phase. All right, which of the following processes is most responsible for getting genetic information into each cell in the body of an organism? Okay, so hopefully you picked mitosis because body cells are made in mitosis. And so when mitosis occurs, it makes sure that your DNA, your genetic information, gets correctly put into each body cell. So the next video will be on meiosis and sexual versus asexual reproduction.